Hi everyone, I'm Michaela and I did my senior research paper on the biodiversity of earthworms and ecosystem services in soil. So for an overview of what I'll be discussing, I'll begin with explaining what ecosystem engineers and ecosystem services are and why biodiversity matters in this context. Then I'll briefly mention two different methods of biodiversity calculations, and after that I'll be going over the three most common methods of earthworm classification and how these relate to biodiversity calculations. To begin, ecosystem services are things such as nutrient cycling, flood control, water storage, and climate regulation that are provided by ecosystems. These services can be viewed as a connection between a healthy ecosystem and the benefits this provides to humans. As I'll explain later, these services depend on other factors such as the health of an ecosystem, as well as the biodiversity within that ecosystem. Ecosystem engineers are organisms that regulate the resources that are available to other species, and they do this by causing physical state changes of materials within their particular ecosystem. So, soil invertebrates are considered ecosystem engineers within the soil. They're a very diverse group of organisms, and they're thought to make up to 23% of all identified species. Of these soil invertebrates, earthworms can be viewed as one of the most important ecosystem engineers due to the fact that they play a huge role in nutrient cycling, climate regulation, plant growth promotion, soil formation, and much more. Perhaps the most important point surrounding this connection between ecosystem engineers and ecosystem services is biodiversity. Earthworms are involved in the provision of these services, but a variety of earthworm species is necessary. This is because within a particular ecosystem, different earthworm species have different functional roles, and therefore are involved in particular ecosystem services depending on these roles. There are a few studies that specifically looked at the status of an ecosystem and its services when there were earthworms present versus when there weren't. Clemens et al. Found that the removal of earthworms in a particular ecosystem resulted in a decrease in soil moisture and infiltration rate, as well as a decrease in soil porosity. Another study also found that in the absence of soil macrofauna, such as earthworms, infiltration rate was decreased, while when certain earthworm species were reintroduced, infiltration rate began to increase again. And Ehlers found that after 10 years of earthworm activity in a particular ecosystem, water storage in the soil was 25% greater. So it's not only important for this concept to be understood in general, it's also important that biodiversity is quantified in some way. For example, to raise public awareness of the value of a particular ecosystem, show the functionality of an ecosystem, such as a biodiversity resource, water storage, or nutrient cycling resource, for example, and to provide an area that can serve to monitor agricultural or industrial effects on the soil. So to quantify earthworm biodiversity then, there's different indices that may be used, and the two most common ones are the Simpson Index and the Shannon Index. Perhaps more importantly though, individual earthworms must be counted in order to calculate biodiversity using these indices. This requires some sort of classification method of the earthworms though. The three most common methods of earthworm classification are morphological, barcoding, and functional. Morphological classification divides earthworms into species and/or families based on physical features such as body color, body length, segment number, and the shape, length, and position of the clitellum, which is part of the earthworm that secretes the cocoon during reproduction. This method is effective since it's pretty simple to do and it doesn't require much equipment. Classification based off of barcoding is another method that may be used to count earthworms. Huang et al. Were the first to study the sequence diversity of the mitochondrial cytochrome C oxidase 1 gene in earthworms in order to determine the species of 86 individual earthworms. The earthworms were then placed into 30 species and 3 families, from which a phylogenetic tree was able to be generated. So this method allows for more precision compared to morphological classification. However, in the paper, the authors noted that 100% of their results agreed with what species each earthworm would have otherwise been classified as based on morphological method. So these two classification methods may be useful complements to one another then. Lastly, functional classification is a method that appears to be mentioned the most within scientific articles about earthworm classification. This system of categorization divides the earthworms based off of habitat position within the soil, the principal components of their diet, and a few morphological characteristics, including size and pigmentation. Using this system, an earthworm is either anisic, endogaeic, or epigeic. Epigeics do not form burrows and therefore spend their lives feeding on the rich organic litter on the surface. They're 5 to 15 centimeters in size when mature, and they're bright in color. Anisic species are deep burrowing in the mineral and organo-mineral layers down to a depth of 1 to 2 meters, thereby aiding in soil porosity, aeration, and filtration. They also aid in nutrient cycling and litter breakdown by mixing of the surface litter, which is their primary food source, with the lower soil material as they burrow. These worms are 15 to 20 centimeters in size, and they have anterior-dorsal pigmentation. Endogaeic earthworms live and feed within the soil as well in the mineral and organo-mineral layers. However, they form horizontal burrows, which also aid in porosity and nutrient release. They're 5 to 10 centimeters in size and unpigmented. You can see these morphological as well as functional characteristic differences in this figure, which shows a representative of each type of earthworm along with their typical burrowing pattern within the soil. In terms of biodiversity calculations, this method of functional classification may be especially useful since it's been noted that ecosystems containing all three of the functional groups are better functioning in terms of biological and chemical processes, as well as in contributing to ecosystem services. So this method of functional classification may be useful for biodiversity calculations. However, it's not necessarily as simple as the other two to perform. So typically, in order to place an earthworm among one of the three categories, observation of the earthworm's feeding habits, microbiological assessment of gut contents within the worm, or food intake and growth rates of the worm have to be studied. Briones et al., however, were the first to use a new technique to determine earthworm functional classification, and this was carbon-14 dating. In this study, analysis of the carbon-14 within the worms was used to determine what role they play within the ecosystem, and therefore their functional classification. Since these functional groups divide the earthworms based on what they're ingesting, the age of the assimilated carbon within the earthworms can be used to determine which stratum of soil each worm is feeding in. So in other words, different functional groups feed on different ages of carbon. The results of this study ended up supporting the claims that epigeic worms ingest plant material that's more recent in the soil layers, since they inhabit surface layers, and that endogaeic species ingest older and more complex material, such as humified matter, since they inhabit deep soil strata. And you can see this by looking at the calibrated age column on this chart for each individual worm and comparing it to the collection date. So here, the species L. rubellus is epigeic, because you can see that the carbon they feed on is more recent in the soil layers, while A. kilinginosa is endogaeic. The carbon they feed on is from older material in the soil. The anisic worms, however, which were thought to typically feed on material closer to the surface, were actually found to assimilate older material similar to endogaeic species, but this is probably due to the fact that anisic worms are known to be more variable in their feeding habits, especially between development and maturity. So when all this is taken together, it seems that all of these methods of earthworm classification are useful when used in some combination. So for some conclusions, humans depend on these services such as water storage, flood control, plant growth promotion, and nutrient cycling, but these services overall depend on the biodiversity of species within an ecosystem. Biodiversity of earthworms in particular results in a variety of functions being performed by different earthworm types. As was previously explained, these services are less beneficial and productive in the absence of a variety of earthworm species. If the functioning of a particular ecosystem is to be quantified for the purposes of land and species conservation, agricultural planning, or pollution control, earthworms must be counted in some way. Because of this, it's necessary that a uniform process of earthworm classification be established. And then once this is done, it will be useful to have data comparing the biodiversity within an ecosystem to the health and functioning of that ecosystem. So although this area of soil research is still developing, it holds the potential for valuable discoveries in the area of soil health, the connections of biodiversity and ecosystem health, bioindication, and much more. These are my references, and thank you for listening.